Well, thank you all for coming for this hybrid Grand Rounds event. I hope this topic will kind of interest people and stimulate a little bit of interest in translational research. I'm not a basic science background, by training, but I've kind of picked up those skill sets during residency, a little bit in medical school, and then post-residency. And I think this is a real passion of mine. It's really changed the way I think and how I think about neurosurgery. And so this topic is really dear to my heart, and I think hopefully it'll interest and stimulate other people to have a similar interest. This topic is on endogenous retroviruses and gliomas, which is kind of a very niche topic. I think it's very interesting, and so hopefully we can get some interest and questions from the audience. If you guys want to interrupt me on any slides that don't make any sense, please stop me. So I'm going to jump right in. Glioblastoma, we all know, is just a terrible disease. I obviously don't need to tell this audience here, but recurrence is really inevitable in every single patient, no matter what you do, no matter who you are. If you're Jock Morkos, Rick Komatar, you resect the tumor, you give chemoradiation, and the tumor comes back. Carolina Benjamin, exactly. And no matter what you do, the resection, these tumors come back. And we've been trying to make a little bit of movement, but in the last 20 years, survival curves have really not changed much. And so really the field in cancer research has moved to kind of multi-omic studies. And if you look at every single paper in Science, Cell, Nature, every single one have these beautiful little maps of, single cell and bulk RNA-seq kind of transcriptomics, proteomics, epigenomics kind of readouts. And the idea is that we're hopefully trying to capture different transcripts that maybe guide treatment-based approaches. I mean, the idea is this idea of personalized medicine, guiding our treatments towards these, transcriptomics or proteomic approaches. But my interest has really been in the last kind of untapped omics, which is the virome. And the WHO estimates that 10% of cancers are kind of linked to viruses, but no such link has been seen in gliomas or brain tumors really in general. But as you can imagine, we know that different types of Epstein-Barr viruses can cause different types of cancers. HPV is the most common one we know that causes cancers. HTLV, human T-cell leukemia virus, which is also a retrovirus, has been linked to leukemia. And so there's a lot of different types of viruses that are associated with cancers, but no causative link has really been shown in glioblastoma. And there's been a few studies that people are aware of, with EBV and CMV, but those have not really come to fruition. And so the question is, are we missing something? And is viral exposure like an epidemiologic risk factor? And so, I'll say this, the retrovirus is really often neglected in large-scale viral oncology studies, because most of the time people are focusing on these large DNA viruses or large RNA viruses, and they really neglect the retroviruses because they're not necessarily pathogenic into the brain. And so this kind of gives me back to a little history, which I think is quite fascinating. In 1920, this man, Peyton Roos, discovered really the first retrovirus. This was avian, called Rue Sarcoma Virus, which is now it has a different name. It was the avian virus. But essentially what he did was he took tumors from chickens, isolated them, ground them up in a blender, basically grabbed the supernate, treated it with the antibiotics, filtered it out so there's no cells in it, and then injected that supernate into other chickens. And guess what? They all got tumors. So he said, what is in this substance that's causing tumors in these chickens? And he didn't know. He kind of just published his papers in the 1920s. This was before the age of, RNA and DNA were even known. So he kind of published his papers, and that kind of went along for another 40, 50 years, until someone realized that if you took this virus and you transformed some cells, those cells still maintain the same exact traits and genetic and genes as the predecessor. So these were kind of inherited in a Mendelian fashion. So what does that mean? That means the virus is not a transient thing, it's integrating into the genome. And so that kind of brought this idea of RNA tumor viruses. And so, this kind of led to the Nobel Prize being awarded to David Baltimore, Renato Delbecco, and Howard Temin in the 1970s for their discoveries. David Baltimore found reverse transcriptase. This was in New York. In fact, there's a couple of prominent researchers here in Miami who actually trained under David Baltimore. Howard Temin actually died before he could get his Nobel laureate. He died of lung cancer from smoking, which is kind of interesting. But this whole story of how they all came together, and they basically found out that discovered the retrovirus at the time when HIV was becoming a pandemic. So it really was a very kind of happenstance discovery. But this kind of led us to the field of what modern day retrovirus is. And this kind of, over the next 10 years, it became discovered that in our genome, as we started to sequence our genome piece by piece, we realized that 8% of our genome was formed of these retroviral elements. So what does that mean? That means that every single person in this room, 8% of our genome have these kind of sequences: GAG, Pro, Pol, Env kind of sequences in our genome, 8%, but most of the time they're not doing anything. We're not out here shedding retroviruses normally, but these sequences are in our genome, and they were kind of acquired as we evolved in our germline. So essentially, while we were monkeys and apes, we picked up these retroviral infections in our germline, and it kind of went, and as we evolved, we all tended to have these same sequences. So really, what are they doing? There's different classes of HERVs. The most common class, which is the most recently integrated, is the one called HML2, which I'm going to kind of go over, which is part of the HERV-K family. And they all kind of, they can all code, and they have open reading frames for these proteins, such as envelope, GAG, reverse transcriptase. And so you can actually see these sequences in our genome, which is quite interesting. So the question is, are these just junk DNA? They're 8% of our genome, but I'm telling you right now that we're not infecting anybody here. When you go and touch somebody, not giving them a retrovirus, or we're not shedding them. So are they just junk DNA? And this is kind of interesting because a lot of the research you hear, and when you hear in Grand Rounds or other places, they're focusing on genes that encode proteins, and that only forms about 2% of our entire genome. Repeat DNA, or even HERVs, form 8% of our genome. So 4 times the amount of protein coding region are formed by endogenous retrovirus sequences. And 60% of them are formed, are these kind of transposable elements. So either DNA transposable elements or RNA transposable elements. Or retrotransposons, as we described. So really, we're ignoring about the majority of our DNA by focusing on these genes that encode proteins. So I think it's a very fascinating field to look into, and there's a push now to look into these elements. And so what I'll say first is these elements are not really junk DNA. They actually have a physiologic function. So if anybody remembers from physiology, syncytiotrophoblasts of the placental development, that entire protein, syncytin 1, I don't know if anybody remembers that protein, but that was from endogenous retrovirus W. So HERV-W envelope protein is actually the syncytin 1 protein. So during embryogenesis, these proteins actually come out and they start being expressed, and probably to allow the stem cell state and allow for embryogenesis. And then as the embryo develops and differentiates, these proteins actually go away. And you'll see this is kind of the theme of the talk today and kind of what we're looking at. So there's a lot of different retroviral proteins that exist and are being produced in our body, but they're very tightly regulated. So what does that mean? That our body is actively trying to suppress these retroviral proteins from being expressed, and they do it through different types of proteins. There's actually a complex called the Hush complex, which silences the retroviruses, and there's DNA methylation, chromatin remodeling. These are all different epigenetic factors that silence these proteins from being expressed. But we all know cancers have a marked epigenetic dysregulation. So in many cancers, these proteins can be re-expressed because of different issues with chromatin remodeling and histone methylation. So this kind of brings me to the next point, that these retroviral elements are very highly expressed in a variety of cancers. And so this was kind of noticed in the late 1990s. But I'm going to start this kind of talk a little bit about koalas. Koalas are very cute animals, but there actually is a very interesting case study, because in Australia, what's happening is there's actual active endogenization of a retrovirus, meaning we're watching in real time koalas get a retroviral infection, and that's integrating into their genome and spreading within the population. And this happens over a course of 50,000 years, but we're able to witness this in real time. And what's happening is in northern koala, there's areas where they're not having endogenous retrovirus, or don't have HORVs by, say, koala endogenous retrovirus. In the South Australia, they're actually having more endogenization. And so what's happening is the cancer rates in the South Australia cohort are much higher than the ones in the North Australia cohort. This was published in Nature Communications maybe I think last year. And so they're noticing that these endogenous retroviral sequences are actually inserting into different genes, such as tumor suppressor areas, or inserting to activate oncogenes in these koalas. So this is kind of a real-time, case study watching how these endogenous retroviruses are causing cancer in koalas. So then the question is, how are they doing it? And so I can, these endogenous retroviruses can do it in several different ways. One is they can actually cause insertional mutagenesis. They can insert into tumor suppressor genes like P53 and prevent them from functioning. They can come as a promoter in front of oncogenes and actually act as a cis activating promoter. So activating the oncogene to make proteins, or the direct protein products themselves can actually activate different cellular pathways such as AKT and Notch. So these are, several different ways in which these retroviral elements can be oncogenic. And so if you take tissues from different patients like breast cancer, endometrial cancer, colorectal, you can see that there's overexpression of different HERVs elements. The most common one is this HERV-K, which I'm going to get back to, but these are really overexpressed in a variety of cancers. This is a paper published in Nature Genetics showing that ERV1, which is a very ancient retrovirus, was a predictive biomarker for prognostic biomarker for survival in kidney cancer. And if you look in gliomas, not really much has been discussed. This is diffuse gliomas. And you can see HERV-K has some mildly elevated expression, but nothing really substantially written about it. And the literature for gliomas and retroviruses, if you go on PubMed, you get 8 hits, and 4 of them are from our lab. So really only 2 articles out there. And one of them is this article that basically suggested that, the HERV-K really doesn't play an important role in glioblastoma. And so they just looked at RNA values and they said, okay, well, HERV-K is only expressed in, 15 to 20% of these tumors. Yet another study came out just maybe a year ago suggesting that these retroviral elements are widely expressed in gliomas, but this was really not an in-depth study that was performed. So, not a lot is known in gliomas, and so why is that? Why do you think? I mean, we have so many big, atlases for gliomas. We have the Cancer Genome Atlas, we have the Chinese glioma atlas. Why aren't we capable of classifying endogenous retroviruses? Well, the fact of the matter is, most of the time, these people, we are ignoring these sequences completely. So if the TCGA and CCGA actually don't even annotate for any of these retroviral elements. So they annotate for 2% of the genome, but the other, these other 8% of HERVs or the 60% of transposable elements are not necessarily annotated for. And when you do your conventional bioinformatic pipelines, you don't account for them at all. So if you try to look in the TCGA for any of these HERVs, you maybe find 4 or 5 transcripts, but there's over 30,000 transcripts in our genome. So we're missing quite a bit. And so if you really want to find these HERVs, you have to do a custom pipeline to identify these elements. You have to prepare your DNA and RNA in a specific fashion. And only in that way can you actually detect these transcripts. So so I'll stress this, a custom kind of pipeline is necessary. And so this is kind of what we're employing in our lab. So we kind of started with an exploratory analysis. We used a public database, ENCODE, which actually has very robust RNA-seq data, but they only had really 4 glioma cell lines. So you have astrocytes and different glioma cell lines in varying degrees of aggressiveness. A172 being the most aggressive, kind of most proliferative type of glioma. And we're able to kind of apply this, methylation analysis, RNA-seq, and kind of, put them together to see whether or not these HERVs are being overexpressed in the glioma cells compared to astrocytes and vice versa. So we took kind of an unbiased approach, bioinformatics. There's a lot of work done by one of our medical students, Vedya. And so this is the pipeline we use, Telescope. We had the help of Corey Johnson, who's the kind of bioinformatic lead at the NIH. But essentially, we're able to identify, these are endogenous retroviral elements in gliomas for really the first time. And you can see that in the cells that are the most kind of indolent. At the bottom here, you see there's very low expression, of endogenous retroviruses, and the cells that are most aggressive, you can find that there's much higher expression, and especially this one element, HML6, and that's also part of the HERV-K family. So this was kind of interesting to us. We noticed that there's one that's really, really widely expressed in the A172 cell line. So we said, okay, well, what does that mean? What is this element doing, and what's this kind of functionality? And so we looked into literature, and again, there's 2 papers on HML6 in the literature. One of them was this paper that kind of did a comprehensive characterization for that. And we noticed that the one in chromosome 19 was the one that we actually were found that was overexpressed. And that one actually encodes for a protein. So I said, okay, great. We found a transcript that encodes for a protein that's overexpressed in glioma. Okay, well, that's great. This is all kind of computer work, but does that mean anything in real life? So we generated some primers, and we confirmed, okay, well, the RNA is also overexpressed in the glioma cell lines as well. So we took patient-derived glioma cell lines and established glioma cell lines and showed that the RNA was much higher in the GBM cell lines than the normal astrocytes. And then we confirmed and did Sanger sequencing to confirm that that was actually within our chromosome 19. We were able to publish this in scientific reports last year. And then, the important thing is, okay, well, can we visualize these RNA sequences in the genome? So basically, this is kind of a newer technique called RNA in situ hybridization, or RNA scope. But essentially, you can probe the RNA elements by having a specific primer to the RNA, and then kind of conjugate that to a fluorophore, and you can actually visualize individual transcripts within cells. So these red dots here, you can see it's kind of blurry here, but the red dots are individual RNA transcripts for the endogenous retrovirus element K. So these are HML6 transcripts within glioma cell lines. So the first time we're able to visualize the RNA in these cells. And so that's great. We kind of showed, okay, well, now we can see the RNA. We see the kind of the full change expression in silico. So we have the computer model, we have the RNA, but then we know this actually encodes for a protein. So some of my mentors at NIH said, okay, why don't we make an antibody against this protein? So we went out and contacted the company, we gave them the transcript, we made a plasmid against that transcript, and we inoculated a whole bunch of rabbits with this plasmid. They generated an antibody against this protein. So an antibody against HML6, and we validated that, in a Western blot to kind of show that this actually matches and correlates really well with our qPCR data. So this is the protein kind of configuration if you use these kind of online, UniProt databases, but you can actually see that this is like it has an alpha fold here. It's about 11 kilodalton. So a very small protein, but we really don't know what the functionality of this is. So then the question is, okay, well, that's great. So now we have this protein. So next thing is, okay, well, is it overexpressed actually in glioma cell lines or glioma patients? So we took some epilepsy tissue as control and we stained it, and you can see there's very bland expression, not really much at all. And if you look at GBM tissue, it's really much higher expressed. You can see here robust overexpression in IHC staining for this HML6 envelope protein, and you can see here it's really highly expressed in this area in GBM. And this was this in several, like 8 or 9 patients that showed overexpression of this HML6 envelope protein. So then we also went into TCGA, and I said there's about 6 or 7 transcripts that are actually encoded in TCGA. Luckily, this one transcript is actually somehow encoded and actually annotated in TCGA. So if you actually look at the TCGA database and look for expression of this one locus, that actually correlated to worsened survival in both aggregate GBM populations and wild type GBM populations. So this is again, independent of IDH mutational status. And if you look at CCGA, this is again maintained as well in the Chinese Cancer Genome Atlas, where the overall survival is worse in patients who have a higher expression. And interestingly, we looked at our RNA-seq population from the University of Miami, and again, in a third independent data set, it's also a worsened survival. And so here you can see that the IDH mutant status has less HML6, and that's kind of enriched in the classical GBM phenotype. So then, this is some new work we're doing in the lab, but essentially you can take siRNA and knock down this transcript specifically. So we're actually knocking down HML6 here in glioblastoma cell lines. And this is kind of suggesting, okay, well, what is the actual function of this? And, I did not want to go into too much molecular biology here, but we're actually generating some really interesting data on the actual molecular function of HML6 as a potential oncogenic protein for proliferation and invasion in GBM. So then the question is, so I'm going to kind of switch gears now from HML6, which is kind of a newer, kind of novel retroviral element, to HML2, which is the more kind of known, more published retroviral element that's in gliomas. HML2 is widely expressed in breast cancer and leukemia cell lines, as I mentioned, but in gliomas, really nothing has really been written. So we did RNA scope again in HML6 for HML2 in glioblastoma. And here you can see, there's differential expression here in glioblastoma cell lines. This is now we're standing here at the top is actually HML2 envelope protein in GBM, and the bottom is the RNA scope. So both RNA and protein here. And, different cells have different RNA experience, different RNA exposure. So here, this is RNA scope for in a GBM cell line, different types of retroviral transcripts. Other cells have other endogenous retroviral transcripts. So, some are HML2 predominant, some are HML6 predominant. So really depends on the cell line and its overall expression. But I think each one of these proteins actually makes and contributes to a different cellular phenotype, and I'll kind of get into that later. So again, that's great. Anything you do in cells, you have to make sure that's clinically validated. So, even if I show something in a petri dish, that's great, but you have to validate that with patient tissues. So we took once again control tissue, epilepsy. So we use mesial temporal sclerosis as a control. This is staining for HML2 envelope protein. Really no expression of HML2 envelope in epilepsy. But when you stain for GBM tissues, this is multiplex immunofluorescence staining with GFAP and HERV-K, and you can see kind of robust retroviral protein expression in GBM cell lines, in GBM patients, excuse me. This is a 70-year-old with this kind of right frontal GBM. Again, here you can see the yellow is the retroviral envelope protein expressed in these patients. This is just a variety of patients with heterogeneous expression of HML2 envelope protein here on the left. And if you take CSF from these patients, so you take CSF from the peritumoral region and CSF from the peri-epilepsy region, and you take them and you do digital droplet PCR, you can actually show that the RNA values in the gliomas are higher than the controls. And this is maintained in CSF and also in the tumor specimens if you digest the RNA itself. So again, the RNA is higher, the protein looks like it's higher as well. And so it is differentially expressed in GBM. Now, that's great. So we kind of show that the protein is there, but, it really kind of the field, really you have to validate this with, with bioinformatics and these kind of bulk RNA-seq approaches. So we were able to get a cohort of about 75 patients from Miami. We were able to get them deep sequenced at the New York Genome Center. This is kind of deeper sequencing, like 4 times deeper than the TCGA. So TCGA is 25 base paired, really non-pair ended data sets. Our database is 100 base pairs, paired end, and ribosomal depleted. So really, once again, as I mentioned, you can't use the Cancer Genome Atlas, you can't use the Chinese Glioma Atlas or these other cohorts because they're not annotated for HERVs. So luckily, our RNA-seq data set is, and so we're able to do kind of really in-depth, bulk RNA sequencing. This is just kind of we made this yesterday, but essentially you're able to generate a heat map of every single HML2 loci. So there's over 100 different loci of HML2 in the genome. These are all of them kind of differentially expressed for different patients. So this is like 75 patients here with differential expression of HML2. And you can see, the kind of as they're being expressed in each subtype. So the new VRHAC classification. So for those of you who don't know, we hired a new kind of head of basic science research, Antonio Ivorone, who kind of reclassified the system for GBM classifications for their molecular subtypes. And so here you can see on the left the differential expression of those within it correlated to HML2. And we have a lot more figures. I'm not going to go into detail about that, but you can see that the patients are clustering based on their HML2 expression. So here, in these red, you can see clusters of patients with high HML2 expression. So really, what does that mean, and what are the next steps? So that brings me to the next point. What's the functional role of HML2? What's the functional role of these retroviral proteins in gliomas? And this is super interesting, very fascinating. This is a paper from Nature in 2014 by Grow, basically suggesting that in the blastocyst, so in our 8-stage pluripotent state, you can actually see retroviral proteins being expressed from the blastocyst on the surface. So here you can see these little retroviral element, viral-like proteins being produced from the blastocyst, and they're highly expressed with HERV-K envelope, so this HML2 envelope, and they suggest that this envelope protein is essential for OCT4, which is a pluripotent transcription factor expression. And interestingly, they found a area within the HERV-K locus that actually promotes that OCT4 binds to. So OCT4 binds onto HERV-K and promotes the envelope protein formation. So this is kind of a bidirectional role of HERV-K and OCT4, and this is published in Nature about 6 years ago. But this has kind of laid the foundation for the role of HERV-K in cancer stem cells. So our lab at the NIH again, looked into this, and they realized that the HERV-K acts via the mTOR pathway to prevent differentiation of neurons. So essentially, through HERV-K, their focus is on more neuroscience. So they're looking at ALS. So they think that HERV-K activation in ALS kind of drives neurons to de-differentiate and try to divide, which forces them to die, and hence cause neuronal damage. Versus in cancer cells, whenever they're activated, you end up having a more pluripotent de-differentiated state. So you can see kind of two sides of the spectrum. But this is kind of important work done, published in PNAS in 2020. And again, this field continues to be interesting. Another publication just maybe 6 months ago in Cell Stem Cell, suggesting another mechanism of how HERV-K disrupts cortical patterning and neuronal differentiation through NTRK3. So, I'm basically harping on the stem cell thing because I think it's very important in gliomas. So we kind of looked into this in gliomas a little bit about different ways that it can contribute to a stem cell development in gliomas. And so this is kind of the really important work here. So now we're staining for about 11 different stem cell markers here in GBM. So here we're staining for OCT4, SOX2, Nestin, and Vimentin at the same time, along with HERV-K. And you can see there's an area here which is kind of enriched in stem cell markers and the area which is not enriched in stem cell markers. This entire slide is GBM, but there are certain areas which are stem cell-like and certain areas that are not. So this is what I call the stem cell niche. And you can see HERV-K is really highly expressed in those stem cell rich areas in GBM. And this is not only in this is like I don't know what happened to this slide, but here you can see that the cellular cells that are enriched in these stem cell markers are also enriched in HERV-K as well. So then that's great, but then can we show that individual cell populations are also the cells that are more progenitor-like? Are they also enriched in HERV-K as well? So then we did single cell RNA sequencing of glioblastoma, and we took publicly available data and kind of looked and see whether or not in different subtypes of GBM, whether or not there's enrichment of HML2 in different subtypes. So here you can see on the left, it's kind of hard to see, but this is you see astrocyte-like cells here, which are in red, mesenchymal subgroups, which are kind of over here, and then the neural precursor cells, which are kind of the most de-differentiated pluripotent kind of like cells, are here at the bottom. And here are different loci of HML2, which are really highly expressed at the bottom in this neural precursor group. And you can see that really nicely here, that the average expression of HML2 is much higher in the neural precursor cells compared to the other more differentiated cellular states. So this was a very small cohort of cells, but we're trying to replicate this in a larger group as well. But this kind of gives us proof of that concept that this contributes to the stem cell niche. Again, you can use CRISPR and RNA interference to knock down HERV-K in glioblastoma neurospheres. So you can actually use this DCAS9, which is an allosteric inhibition of the promoter, and you can actually see neurosphere formation markedly drops off in patient-derived neurospheres. And then, we can also use RNAscope to actually visualize the RNA transcripts coming down. So once again, if you take the CRISPR plasmid and infect these cells, you can see the HERV-K transcripts drop down. OCT4, which is a stem cell marker, drops down. Nestin as well also drops. So this is kind of proof that you can actually visualize disruption of these stem cell transcription factors with HERV-K disruption. And here at the bottom, we just validated that with qPCR. So then the question is, okay, well, what is HERV-K doing? So can you actually isolate, can you take HERV-K and transfect some cells and see what happens? So we transfected some glioblastoma cells with the HERV-K consensus plasmid, and we noticed that OCT4 was slightly elevated in these glioma stem cells. We also did the same experiment in astrocytes and actually showed that OCT4 also dramatically goes up in astrocytes, and they actually become more dedifferentiated as well. We also did CHIP qPCR, which is kind of a way to see whether or not whether or not there's transcription factor binding sites in certain areas, and we found that in the glioblastoma cell lines, there's actually an OCT4 binding site on the HERV-K LTR. So that it kind of gives us an idea that there's a bidirectional interaction between HERV-K and OCT4, which contributes to pluripotency in GBM. So that's great. So now we kind of found out, okay, well, there's some molecular basis for HERV-K contributing to pluripotency in cancer stem cell state in GBM. Can we, take this to an intracranial orthotopic model? Is there any type of survival benefit if you knock down HERV-K in glioma cell lines in glioblastoma? So we took our plasmid, we transfected some cells, and then we waited for them to recover, and then we injected those cells into the right frontal lobe of mice and basically assessed for survival. This is kind of a crude way of knocking down HERV-K because it's a transient transfection, so it's not permanent. But we were able to see a marked survival benefit in the mice with our transfection. You can see that every single tumor that grew, so this is a transient transfection, only about 50% of cells were transfected, but the cells that engrafted were all positive with HERV-K. So essentially, HERV-K was driving, the cells that actually made the tumor. So this was interesting. This was in the controls and treated group. So it would be interesting would be to do a kind of, a spatiotemporal knockdown of HERV-K in situ and see whether or not the tumors are even engraft at all if you actually have local regional control of the transcript. So then that brings me to the next step. HERV-K is an endogenous retrovirus. This is we showed you in the blastocysts they're actually making viral like particles. Is that the case in GBM? Are we just seeing RNA? Are we just seeing some protein in the cytoplasm? What are we seeing here that's actually being produced? And so some work done by one of my colleagues in ATRT, which is a pediatric tumor we all familiar with, showed that there's some retroviral budding in these ATRT cells, and in GBM we isolated some extracellular vesicles, and they are also stained for HERV-K. So you can, in the extracellular vesicles, you can actually find envelope protein in the vesicles as well, which led us to basically doing other assays. So we can actually assay for reverse transcriptase in the supernatant. So you actually can take the supernatant of cells and supernatant of plasma from Dan, for example, and you can stain it for reverse transcriptase. So you can actually do something called a PERT assay. They do this all the time in HIV patients to check to see whether or not the HIV burden is high or low in patients who are being treated. But you can actually do the same thing in humans with plasma. So you can actually take their blood or plasma or CSF and do a PERT assay and assay for the reverse transcriptase levels in each patient. And it's been shown to be a prognostic marker in breast cancer, and in glioblastoma you can imagine the same thing we found. Cells that had high HERV-K had the highest amount of reverse transcriptase. Cells that had low HERV-K had lower amount of reverse transcriptase. And so you can actually target these reverse transcriptase levels with antiretroviral drugs. So reverse transcriptase inhibitors can actually target the reverse transcriptase levels, and this is shown by our lab in this paper on retrovirology. And so that kind of got us to doing some electron microscopy to see whether or not these envelope proteins are being produced on the cells. So we basically, took a gold antibody and conjugated it to the HERV-K envelope protein, and basically you can see here, protein expression on the surface of GBM cells and kind of going out into the Golgi of HERV-K envelope protein. But the morphology here in these slides were not that great. So I said, okay, well, we got to do something a little bit better. So, oh man, this is not the right images. Okay, well, this is the kicker I really want to show you guys, but you can see, all right, well, you have to take my word for this, but they're essentially, you can actually see you can actually see retroviral virions actually being produced on the surface of the GBM cells. I don't have them captured here. I think the slides were corrupted here, but you can actually see the retroviral budding from the surface of the cells. And you can actually see them kind of, if you see here, the very corner here, you can see them. You can see the kind of retroviral element being produced on the surface of these cells. And you can see it's pretty nice looking when it's actually right image. Essentially, abecavir, which is a reverse transcriptase inhibitor, can actually knock down the HERV-K envelope levels in GBM. And so here you can see the HERV-K envelope levels come down with abecavir, which is a HIV drug, and OCT4 also comes down. So you're actually causing differentiation of the GBM cells and reducing their cell viability through using reverse transcriptase inhibitors. So we basically use a system called DepMap, which is a publicly available website where you're able to query the efficacy of different drugs against different cell lines. And if you take every single GBM cell line in DepMap, which is about 30 or so GBM cell lines here, you can see that there's certain antiretroviral drugs are very efficacious. Abecavir is one of them. Lamivudine, which is another reverse transcriptase inhibitor, is also very efficient. Ritonavir and dolutegravir are also very efficient in knocking down and killing GBM cell lines. So here you can see that there's basically 3 of these drugs, and 3 of these drugs are actually found in one pill, and that pill is called Triumeq, and it's commercially available to treat HIV. So the question is whether or not we can repurpose those drugs to for antiretroviral therapy. So, in essence, I'll say that HERV-K is essential for glioma stemness. It's been ignored in the literature usually, but I think that these certain retroviral loci are very important in contributing to the stem cell niche, and it's driven a lot by this LTR5HS, which is basically promoting OCT4 and SOX2. And aberrant HERV-K can really be targeted with antiretroviral therapy. So basically, you can target these sequences with antiretrovirals that penetrate the blood-brain barrier. They're able to be translated to a clinical trial very easily, and we're hoping to do that in the next year or so. And so the future directions for our study are really kind of working on the bulk RNA-seq data to see whether or not there's differential expression of some of these elements in certain cellular states. And then, I want to do local regional HERV expression using these kind of newer techniques. There's newer techniques that Sylvester and the oncogenomics core have, that you're able to localize transcripts. You do RNA-seq, but basically on a slide, and basically look at the transcriptional local regional transcriptional activation of elements within tissue samples. And then, can we make a more targeted approach to target HERV-K? If this is being produced on the surface of cells, can we make CAR T cells against, HERV-K envelope, or HML6 for that matter? And so we're working with collaborators to make a nanobody CAR T cell against gliomas. It's kind of already been patented already. So hopefully we're going to see some good preclinical data in the next few months looking at CAR T cells against HERV-K envelope. And then, can we use this as a biomarker? If we resect a glioma, does the HERV-K levels in the CSF drop off? If you take out a tumor pre-op and the HERV-K levels are high, and then you take the tumor out and sample the CSF where the HERV-K levels are low. So is it a biomarker for treatment response? Can we monitor these levels during treatment and longitudinally? So, there's this initiative, the GLASS initiative, which basically takes, paired dyads of tumors. So basically patients who come in with initial diagnosis and patients who have recurrence, and they basically sequence them at both initial diagnosis and recurrence. So can we take, that RNA-seq data, look at those and see whether or not in recurrence, whether or not, different retroviral biomarkers are higher or lower. And so these are the kind of next questions we're looking at in terms of our glioma projects. We've also done a lot more work in HML6, and we've actually identified some pathogens, some different molecular activators for HML6 activation in gliomas as well, which I didn't want to bore you guys with, so I left it out. But we'll reach out with a talk later on about it. This is our lab, the section of virology immunotherapy. We've been kind of busy the last 2 months. We finally got up and running, thanks to Dr. Dietrich and Dr. Levy and Dr. Lemon as well for their help kind of getting our lab getting up and started. And, our postdoc, Deepa and Jaleesa, have been working really hard to get things working. This is our lab. A lot of thanks to the Noth lab at the NINDS, who's been kind of helping me, mentor me, and giving me some basic science experience. Our bioinformatics lead, Vaidya, who's done a lot of the RNA-seq and bulk RNA-seq data. Deepa, who has really no neurology, neurosurgery background, but has learned a lot in the last 2 months on retroviruses. Jaleesa, who is a post undergrad with us as well, who's applying to medical school soon, and a variety of other people in the lab who are helping out with this. And thanks to our funding from the NINDS, NREF, and the Florida Center for Brain Tumor Research. So hopefully I didn't bore you guys too much.
Great job, Ashish. We're really excited about you starting and looking forward to the future.
Thanks, Dr. Levy.
Patient population dealing with.
Yeah, I think that's a very good point. So the next questions I think for me are to figure out whether or not where these HML2 or HERV-K loci are within these new transcriptional states. So, now that we know that there's a mitochondrial subtype, there's this proliferative progenitor subtype of GBM, where in which of these cells or which of these subpopulations is HERV-K most highly expressed. And then the second question is, if we know that our treatments like temozolomide radiation kind of are affecting the most proliferative cells and they don't affect the cancer stem cell niche, can we add on these drugs to kind of prevent the pluripotent cells, which are responsible for the cellular heterogeneity? Can we add on drugs to attack those cells? So I think that if you're attacking the basis of the cellular heterogeneity, which is the stem cells, and I think you can make a difference in GBM. The next question is, everybody's talking about the immune system now with the, to kind of activate the immune system, and the last part of this, which I didn't talk about, was that HERV-K actually diminishes our immune response, specifically HML2 diminishes our immune response in a similar way with HIV. So in HIV patients, they've shown that HML2 contributes to a lower immune response in patients, so they're not able to, CD4 activation is much less. So the question is whether or not by targeting this, you're also increasing the immune response. So that's a separate project we're actually working on in our lab. So we'll hopefully get to that, but. Yeah, so that kind of gets back to the epigenetic kind of regulation. So the very good question. A lot of these elements are repressed by DNA methylation and histone methylation. So in IDH mutant patients who are hypermethylated across the board, the expression of HML6 and HML2 is much less. And so I don't know if that's, we try to eliminate those patients from our survival curves, but it's very interesting. So I think it's more of a relevant IDH wild type, to be honest with you, because I think there are a lot of these elements like SMARCB1, which is really well known in clear cell meningiomas. If you have a mutation in that histone and the chromatin remodeling protein, then you have higher activation of HML2. So a lot of these epigenetic factors are playing a big role in the retroviral activation.
Great talk, Ashish. Very, very impressive. One question I had is, so if HERV-K is an endogenous retrovirus, that kind of implies that you're getting it from mom or dad. Is there any sort of familial pattern to developing these tumors?
Yeah, I haven't looked into that. I don't think our data set is not obviously robust enough to look at that, and I don't think many people have. They've noticed in one patient population in African Americans who have a certain HERV-K locus that they are more predisposed to developing certain tumors, but not gliomas, and that's because they're inherited, kind of in a Mendelian fashion. So if you have this one kind of single nucleotide polymorphism in HERV-K, then they were more predisposed. That was an obscure paper, but that's, I guess, but there is some kind of ethnic familial predisposition in that sense, but certainly something you could do, I guess, pretty easily. Absolutely, that's kind of what we're hoping to do. So we're hoping to start hopefully a trial where we take patients who are newly diagnosed, treat them with an antiretroviral therapy plus temozolomide. Prior to treatment, take their plasma, sequence for look for HERV-K transcripts. Then after treatment, look at HERV-K transcripts sequentially during each MRI and see whether or not you can correlate to tumor recurrence. My mentor at the NIH has kind of like this pipeline. They already just sample. They have like a robot that does the transcripts for you. So it's kind of a plug and play kind of thing. So it's really nice in that sense. And they actually have the same trial for ALS running with their antiretrovirals, and they had some really nice results in ALS. So the question is, can we kind of replicate the data in GBM? And interestingly, the last thing I'll say is that there's actually now a HERV-K specific reverse transcriptase inhibitor. So a company has just isolated the crystal structure of HERV-K reverse transcriptase, and so that's specific to the cells that are in our human body, not just these NRTIs. So they've actually have a drug that's going to knock down that specific HERV-K. So the question is whether or not we can use that drug as well. So more to come.
Okay, that was fantastic. Just fantastic presentation. And anything you need to get your lab to the next level, please let me know. That's fantastic. Oh yeah, look forward to it. Let me just back up one. Let's see. There you go. Okay, so my name's Dalton Dietrich. I'm the scientific director of the project. I've been at the University of Miami for about 40 years. Scientific director for 25. So over these years, we've really been intimately associated with the Department of Neurological Surgery. So it's great to see what's happening in neurosurgery and how the project can interact with neurosurgery colleagues. So today I'm just going to talk about the pathophysiology and treatment of acute and more chronic spinal cord injury. I gave this lecture a couple of weeks ago in London at an international spinal cord meeting. And I thought I would just expand it into some other aspects that we're doing in the project today. So these are my disclosures. I have a list of them. Most of them not going to affect what I'm talking about today, but I am a managing member of Inflammisone, which is a small company that's producing a whole series of new therapeutics and diagnostics approaches to targeting abnormal inflammasome signaling in CNS injury and repair. So I'll just mention that. Okay, SCI epidemiology, everybody knows about this. SCI is becoming an epidemic in modern society because it's happening all over the United States. We have about 400,000 annual incidents of SCI worldwide, about 18,000 new cases in the US. The average age now is 42 years. Approximately 1.3 million living with paralysis as a result of SCI. Motor vehicle accidents was always the major reason for a spinal cord injury. That's kind of decreasing with age now. It's followed by falls, acts of violence, recreational activities, the leading causes. Males are still more a risk, but females are increasing, as you know. And then we used to talk about the very 2029 age bracket for males. Now we actually have a second or third group up to 70 age group having spinal cord injury. Females are most risk between 15 and 19. And again, we have this 60-year-old group that's increasing. 70% of spinal cord injury victims are due to accidents or injury during serving in the military. So we get a lot of funding from the Department of Defense looking at spinal cord injury. In fact, the Miami project went up to Capitol Hill about 10 years ago and really pushed for more DoD funding for spinal cord injury. Most of the money was going into neurotrauma, but what they meant was traumatic brain injury. So we got a whole separate pool of money for spinal cord injury, which is competitively given out throughout the United States. And currently there's over 5 million people living in the United States with paralysis due to a variety of reasons: stroke, traumatic brain injury, spinal cord injury. So I thought this was interesting slide. The demographics of spinal cord injury are changing. You see these different peaks. You have 3 peaks now. You have this early age that we've been talking about for many years. Now you have this intermittent age, and then you have this age group. So it's really interesting that over time, the motor vehicle accident incidents decreasing, whereas you see a much more increase with the falls and things of this nature. So there are global changes. I think we all agree in terms of remarkable similar in terms of these demographics. Average age is increasing rapidly. This ratio in Asia D to A is increasing. Fraction with early surgery increasing. Motor vehicle accident prevention and severity reduction appears to be very effective. I mean, these cars today are unbelievable compared to what we used to drive. And increased understanding of how to reduce falls. I think it's a major issue. And all these data is coming out from the North American Clinical Trials Network, huge network that's collecting all these data. So what are some of the factors contributing to spinal cord injury from falls? Well, cervical degenerative changes that you guys see all the time, medical states that contribute to falls, reduced balance, weakness, and inadequate home and work environment infrastructure. Spinal cord injury research. This is what we're attacking in the area of research in my project. For example, my interest is primarily in the pathophysiology and trying to understand what type of neuroprotective strategies can we use during the first several days after spinal cord injury. Can we protect? Then, of course, transplantation, regeneration, repair. Can we actually enhance circuit reorganization, regeneration, rehabilitation, neuromodulation? Even if you're lucky enough to produce significant axonal regeneration or axonal sprouting, does that actually produce functional synapses? Does it actually produce an improvement in function? Quality of life. Again, we've been trying to increase motor recovery after spinal cord injury. More and more, it's sensory. More and more, it's these quality of life issues such as spasticity, neuropathic pain, obesity, diabetes, all these comorbidities associated with SCI. Can we then move it into clinical trials? And we're fortunate that we've had over the last several years about 6 FDA approved trials. So this is a center where we can actually take the things to the discovery level and then move it to people. And then can we train the next generation of scientists and clinicians and at the same time serve our community stakeholders? So this is a unique problem that incorporates discovery, translational, and clinical studies. And it's not very many in the world that is doing this. And the only reason we can do this is people like yourself get interested in our science and help us move it forward through this pipeline. So the neuroprotection, it's difficult because the pathophysiology of stroke, brain trauma, spinal cord injury, things that we're studying, it's pretty complicated. You got all kinds of cells that are affected by injury. You've got all kinds of types of cellular and molecular cascades that are activated after injury. So, what do you target? One of the areas that I'm interested in is actually serum and CSF biomarkers. So we can actually look at, the temporal profile of some of the biomarker changes that we see, and maybe we can actually utilize these various therapeutic windows to deliver drugs that actually target what's happening in the person at that time. And then we talk about patient heterogeneity a lot. We heard it just today, but we have a big issue in terms of which patients they look the same. The MRI looks the same. These relatively crude behavioral assessments look the same, but they're quite different when you really dig deeper. So this is an area that we're trying to work on quite a lot. This is a paper just came out. Greg Bickford, he's an outstanding postdoc in the MIMI project, and this is just a review of systemic inflammation after SCI, reviewing of biological evidence related to health risk and potential therapies. And this is a really nice paper that talks about the different strategies that we should be using to target these multi-organ effects after spinal cord injury. This is a study we did with Michael Wang and colleagues and Kevin Wang at the University of Florida, where we're using differential proteomic and system analysis in spinal cord injury. And we've got these beautiful networks of the different types of proteins that are interacting, genes, proteins, and other cell signaling cascades that we're studying to try to figure out exactly what's the best way to approach this therapeutically. And getting back to patient heterogeneity, you can just see with the GFAP here, you can see the heterogeneity of these two patients. Both of them are Asia A, but you can actually see over time, days after injury, you have different types of biological effects happening in one patient versus the other. So patient heterogeneity is issue. So I thank Timur for this particular slide. Many years ago, Dr. Clancy started looking at intraoperative monitoring with Dr. Barth Green, and this is just the somatosensory evoked potential responses that you see during a surgical procedure. So a lot of things happening, as you know of, in the intraoperative stage. I had a neurosurgeon call me one day. We'd just written a paper, the first paper on hypothermia after spinal cord injury. And he said that he was doing surgery. I think he was getting a tumor out of the spinal cord, and all of a sudden the SCPs went flat. So he says, "Can I just pour cold saline on this particular area of laminectomy?" And I told him, I'm not a neurosurgeon, I'm not a clinician, but maybe you could. And he actually did that. He called me up. He said the patient recovered in terms of that particular incident during the surgical procedure. So anyway, so this is new papers that are coming out. This came out in Neurosurgery Spine. let's see, this year. And these authors are doing multimodal interventions to optimize spinal cord perfusion in patients. And this is a big area right now where physiology is really coming back. And this is a systemic review. And let's see, over 2,500 articles are identified and 72 are processed for full text review. Conclusions: this review demonstrates the importance of assessing how different interventions may vary in the ability to optimize spinal cord perfusion pressure. So arterial blood pressure, interspinal pressure, CSPP as predictors of outcomes. So that I thought this was kind of interesting, because in the laboratory, we don't look a lot about physiological variables, and more people are taking this to heart. This is a really good study. I was just in track SCI. You know, a lot of people have heard about the track TBI study, but this is coming out of San Francisco with a lot of the colleagues that we work with, Michael Beatty, Adam Ferguson, and of course, Dr. Manley. And so they've done intraoperative monitoring records, and neurological outcome data were extracted from 128 patients. They looked at machine learning, big data, novel approaches, identification, the importance of predictors and recovery after SCI. Network analysis suggested the time outside the optimal mean arterial blood pressure, hypotension or hypertension. I always knew hypotension was bad because we've been doing that for years in TBI and stroke. But hypertension, they turned out that during surgery was associated with lower likelihood and neurological recovery. And I don't know what the guidelines are at Jackson, right, or what you're doing, but the optimal mean arterial blood pressure is between 76 and 104 to 117 millimeters of mercury, associated with the best neurological recovery. I think if you get the blood pressure too high, I think they hypothesize you get an increase in hemorrhage in these patients. So this is a great example of big data machine learning that's being used in neurosurgery right now. You know about time is spine. This is coming out of Michael Failings. I mean, a lot of people are studying it, but I see more and more papers from Michael Failings looking at surgical depression within the first 24 hours. And understand that in Miami, you guys, when it's appropriate, you're trying to decompress as early as possible. This was kind of interesting that in the first 24 to 36 hours following SCI, there's a steep and continuous decline of motor recovery with delayed surgical decompression. After the first 24 to 36 hours following injury, motor recovery plateaus and the efficiency associated with early decompression is lost. And in cervical SCI, the additional improvement in total motor scores with early decompression is greater in the upper limbs or just below the level of injury than in the lower limbs. So there, these, again, big data sets are allowing us to really dissect out exactly when it's best to decompress and what the effects are. And this is just pooled analysis. And again, I don't know why this keeps doing this to me. Surgical decompression within 24 hours of acute SCI is associated with an improved sensory motor recovery. And the first 24 to 36 hours after injury appears to represent crucial time window to achieve optimal neurological recovery with decompressive surgeries. Okay, now that I've kind of got out of the surgical realm, we'll go into the neuroprotective treatments. And there's a whole list of drugs that have been used internationally. And so far, none have really made the grade in terms of being protective in big multi-center trials. This is a review, again, by Michael Failings and colleagues. The three drugs that they actually talked about in this review for neuroprotection is Realizol, systemic hypothermia, and G-CSF. So I thought I would just briefly talk about that. A colony stimulating factor for spinal cord injury. It's been around for a long time. There's a lot of preclinical studies showing efficacy. This is a phase 3 study showing no significant difference in the primary endpoint between G-CSF and placebo, but I read that there are additional studies going on in the world to actually evaluate this particular therapy. Realizol, you know all about that, a sodium channel blocker, but now it's this excitement that it actually targets some of the excitotoxic mechanisms, cell signaling cascades. And of course, Dr. Grossman, he was a good friend of mine for many years, and he just, really formulated this North American clinical trial network, and it was really the guiding light to these studies. So the Realizol design, clinical critical endpoints, things like that have been done. This is the results of the phase 1 study for Realizol and SCI. And you can see that one group, this is the Asia B group, actually showed significant improvement, where the other ones really had less effect. But again, patient heterogeneity, how you set up your clinical trials, what you're giving dose and things like that. And just point out that what the studies that are really going on now have to do with pharmacokinetics of Realizol. And this is just a study that came out last year. And because of the complexity of the pathophysiology of SCI, therapeutic doses may change. So I think they're going back to the drawing board about actually doing the pharmacokinetics of something like Realizol, which have been studied for years and years and years. It's like progesterone, right? Progesterone was working really well in many TBI models, and then when it moved to the clinic, it did not work, and they're blaming that also on pharmacokinetic aspects of the injury. Okay, so in our world, replication and reproducibility of the result is extremely important. So yes, someone comes along and shows a positive result with a particular drug in spinal cord injury. The spinal cord community wants that particular treatment to be replicated in another laboratory, possibly with another animal model. Because of course, again, the patient heterogeneity that you're dealing with in the clinical arena, you really need to do that. So this is funding that we got from NIH several years ago. And Ozzie Stewart, Phil Popovich, myself, and Naomi Kleitman, who was at NIH at the time, leading the SCI program, we funded and we tried to replicate some of the most exciting work in spinal cord injury. And I personally found it very difficult to replicate because at first it didn't replicate. Number two, you try to find out exactly what we did differently than the published paper. And many times it was very difficult to do that. So one therapy that's been replicated by many laboratories and many models of acute injury is therapeutic hypothermia. And I wrote the first paper in 1986 on the importance of very small changes in the temperature of the brain in terms of affecting CA1 histopathology, cell death after transient global ischemia. And that took off in terms of the cardiac arrest field. So I just got back from an international hypothermia meeting in the Netherlands, and there's a debate right now in cardiac arrest. What's the best temperature? And I think the best temperature in terms of level of hypothermia is dependent on the patient that you're looking at. There's mild, moderate, severe injury severities and all kinds of types of neurological problems. So if it's a relatively mild injury, I think you can bring temperature to 36. If it's a more severe injury, maybe hypothermia by itself is not going to work. But if you have this intermittent zone, bringing temperature down to 33, 34 that we're doing with Dr. Levy and colleagues on spinal cord injury is probably most important. So it's one of the most powerful neuroprotective therapies that we have. It's beneficial in cardiac arrest, neonatal hypoxia, ischemic stroke. Now they're over in Japan. I just gave a talk, Zoom meeting, and they're thromboectomies. As soon as they're taking out the thrombus, they're cooling the previously ischemic tissue. So that's taking off really nicely. Reperfusion injury, TBI, which we've been working on for many years in spinal cord injury, and it's just not hypothermia. It's called targeted temperature management. That means preventing periods of hyperthermia. And again, we wrote some of the first papers showing that if you traumatize an animal or you produce a stroke and you wait 24 hours and you just artificially elevate temperature to 39 degrees for a few hours, that changed the biology of that insult, and the animals did much, much worse. Okay, so is hypothermia protective in spinal cord injury? This is our first study in severe thoracic injury in rats. And if you wait a couple hours and you bring temperature down to, at this time it was 30 degrees, you can look at locomotive recovery. You see a very nice, robust improvement in locomotive function. So these were the first data really with moderate hypothermia in a model of spinal cord injury. And then when I talk to my neurological colleagues, they go, you know, thoracic injury, okay, but how about cervical injury? So at that time, there wasn't a lot of cervical spinal cord injury contusion models. So we developed one, and this is with my colleague, Dr. Damian Pierce. And you can see from this particular side, we see very nice preservation of the motor neuron population in the cervical cord, and we see very nice improvement in overall upper extremity function. So thoracic, cervical, it seems to work. So what are the mechanisms by which temperature modification can affect outcome? And there are many. This is just a very simplistic slide, but it shows that as you elevate temperature, which happens a lot in our patients, increases excitotoxicity, free radical release, blood-brain barrier breakdown, intracellular signaling cascades, cytoskeletal proteolysis, and then hypothermia kind of blunts many of those things. And it's pretty potent in doing that. And I've spent basically my whole career kind of looking at different types of pathomechanisms by which temperature can affect all of these second injury pathways. We've got a group of microRNAs, for example, that are extremely sensitive to mild changes in temperature. Okay, so this is a systemic review and meta-analysis of therapeutic hypothermia, and one just came out yesterday. So people are still looking at both preclinical literature and clinical literature, and so far it all looks relatively positive. Here's the fever burden. You know, again, the importance of maintaining a normal thermia or hypothermia, or whatever you want to do. And here's spinal cord injury and TBI. In both cases, if you raise temperature, you aggravate outcome in these clinically relevant animal models. And again, these were some of the first data showing that. Okay, so of course, Dr. Levy was very interested in that. Dr. Green had been previously done some hypothermic work in spinal cord injury. So this is the first study that we did, these 14 Asia A cervical patients, cooled for 48 hours, systemic hypothermia. The average time to get to target temperature about 6 hours. And we looked at all of the physiology, heart rate, things of this nature. We looked at, risk of cooling, which is obviously a big problem. We were all concerned about pneumonia and things of this nature. But the data looked pretty good at 1 year, about a 43% conversion from Asia A to B and C. The issue is that, our neurosurgical colleagues that's doing such a great job in the operating room, getting patients in, stabilize them, things of this nature, decompression surgery, physiology. So we don't really know right now exactly, if you don't do anything, what's the conversion rate. And that data is now coming out. So therefore, to really make it and just let you know, we're using these intravascular cooling catheters. I mean, the data is continuously coming out, intravascular cooling versus surface cooling. And we're finding out that intravascular is much more precise in terms of moving the temperature down very rapidly, but even probably more important in maintaining it at a very steady level, and then a very slow increase in temperature. All these phases, bringing the temperature down, maintaining it, slow rewarming, are all critical now. We're finding out more and more as the data is coming out clinically. Yeah. Yes, there's completely two groups that are looking at local versus and trying to figure out what's the best. What is something that you think you could do surgically to actually get cooling to come in and things like that, a local area for many days. Because you've got the Chinese now telling us you have to cool like severe TBI patients for 14 days, for example. So if you wanted to do something like that, how would you do that with a local cooling? But there's companies that are doing that, different devices. And I work with Zoll quite a lot, but with their intravascular catheters. But at the meeting we had about 10 new companies with ways to cool the brain and spinal cord. So it's emerging technology that's really good. Yes.
Another guy tends about over his experience, another major, by the way.
Oh, okay.
60s, 70s, and then they just got abolished. I'm sure they'll have
Yeah, I love talking to Charles Tatter about this. He's got, historically perspective on all of this and things like that. Again, our big contribution, I think, was that it had to do with you don't have to bring temperature down, to 4 degrees C, things like that. That you could probably bring it down in some cases, for example, in cardiac arrest patients, maybe even to 36, and inhibit periods of fever. So it's very subtle in that regard. Okay, so this is the study that Alan and colleagues are running right now, a prospective multi-centered study comparing the complication profile of modest hypothermia versus normal thermia for acute spinal cord injury. And these are the groups. He's recruited, these centers that are fantastic, and the study is going on. Alan can kind of tell us exactly when it's going to be completed, but here's the total. I think George gave me this. The total subject enrollment so far is 60, and you see Miami is leading the pack. So when do you think you'll be able? Is it 100 or 120 patients? What's your target? 120 patients. So 24, 23. 25. Okay, very good. Great. Yeah. But so far, so good, right?
Yeah. It's safety. I published that paper on safety. I published a little bit on the application, but since they're not like
Okay, very good. Very good. We will see. Okay, so what's the next step for hypothermia? So with nanotechnology and things of this nature, we're developing a series of nanodrugs that target receptors in the brain. And in rats, we can now give these intranasally, and you have a nice drop in temperature. And we're trying to do dose response now to see what level we want it to go to and how continuous the hypothermia is. So the vision here is, in the way to the hospital, maybe you could actually induce a hypothermic period, and then you get to the hospital, and the treating clinician can decide whether or not he or she wants to maintain the cooling profile. Okay, so the other area that we're really working in both stroke, TBI, and spinal cord injury is what these injuries do to multi-organ systems. And this is just a review article that came out, and our postdoc, Dean Kurd, 2 weeks ago presented this data to the Department of Neurology, and because we're doing stroke and looking at these multi-organ systems effects and trying to figure out exactly what the mechanism is, affecting the heart, affecting the gut microbiome, affecting the lungs and things of this nature. And our overall hypothesis is after injury, there's these extracellular vesicles or exosomes that are released from the injured brain tissue or spinal cord tissue, and they get into circulation and they circulate all through the body, and they're taken up by different organ systems. And the cargo of that particular exosome contains a lot of things, proteins, microRNAs, and things of this nature, but it appears in our hands to have a large amount of information in terms of the immune response. And so our work is now looking at inflammisone signaling and how that can affect multi-organ systems. This is a paper we published with the group in biochemistry, Sylvia Darnett, who's the chair of biochemistry, and we're looking at the changes in the gut microbiome, for example, and doing a lot of gene sequencing. So my question that I have written down, and we're going to try to approach that in the near future, is if you do systemic hypothermia, for example, in a patient with spinal cord injury, would that actually attenuate some of the multi-organ consequences of SCI? So we'll be looking at that experimentally, and we can also do that, obviously, with various biomarker approaches. Okay, so this gets into the inflammasome signaling. This is an area that we've spent the last 15 years looking at. It's part of the innate immune response, and there's a lot of excitement now in the targeting the innate immune response to different types of viruses, infection, as well as CNS injury. And the inflammasome, as you probably know, is it's a really complex multi-organ complex involved in the activation of caspase-1 and the pro-inflammatory cytokines, IL-1 beta and IL-18. Now, we've been trying to target some of these cytokines, but they're pretty much downstream. And what we want to do is go way upstream where these, the caspases are being cleaved, the activation of IL-1, things like that. And we can actually stop this innate immune response early. And it's really cool that you have different types of inflammasomes depending on what cells you're looking at. And but one aspect that's consistent in all of the inflammasomes that we studied so far is this ASC. ASC is a apoptosis-associated speck-like protein containing a caspase recruitment domain. So it recruits these other types of components of the inflammasome. And so we developed a series of antibodies that block this ASC from forming, and therefore blocks the activation of the inflammasome signaling cascade. So back in 2008, we first looked at inflammasome signaling after spinal cord injury. You see the Western blots there. You give this antibody, and it seems to decrease histopathological damage and improves upper extremity function. So that was kind of exciting. And now, today, move forward, the inflammasome is now involved in many types of inflammatory disease, autoimmune, metabolic diseases, neurodegenerative diseases, secondary injury after spinal cord, traumatic brain injury, strokes, and cancer. And we just did a study with the group in Sylvester on breast cancer, and there seems to be a lot of inflammatory signaling in that. So we're just kind of moving forward. And we had 17 patents, believe it or not, and they all got picked up by Zyversa a couple of years ago. And so they're taking it forward now, and we're kind of watching and doing, and they're funding us to do some continuous work in Alzheimer's disease right now, which looks pretty promising. We just got a new NIH grant on that. And so the future is bright in terms of inflammasome signaling. Glioblastomas, I don't know about the immune response. It'd be nice one day to maybe look at some of those sections, see what's happening. We've looked at ALS with Michael Benatar with biomarkers and really didn't see anything, but now we've got, just found out we have a nice collection of ALS brains in the brain bank. So we're gonna be looking at that. We just finished a big study with ALS and things like that. So we're looking more and more utilizing some human tissue. And with the neurosurgery with Dr. Joe and colleagues, we're now collecting tissue after trauma and looking at inflammasome signaling in the CSF, the plasma, and in the tissue itself. I'm really excited about how that's going. So drug discovery is a big area now, obviously. And I think the project was the first group at the University of Miami to get a high content screening equipment. It was about $500,000 at that time. Vance Lemon and John Bixby came into my office and said, "Do you know anything about high content screening?" I said, biotech companies do that, right? He goes, "Yeah, but why shouldn't an academic institution do it?" So we purchased that, and now they've actually screened, I think they told me, 500 million of compounds now. And downstairs on the first floor, I guess you wonder, what are we doing with all this space on the first floor now that people have moved over to the Lens Center? Well, we're going to university is investing $10 million in state-of-the-art drug discovery program. So it's going to be a fantastic place to be here in the Lois Pope Life Center to do some really cool stuff. Okay, and then finally, repair. This is our focus as well, obviously. It was the reason the project was started many, many years ago. Again, like neuroprotection, you've got all kinds of cells that are involved in reparative strategies. And so what do you target with these types of interventions? Well, the project is kind of divided, axonal regeneration and myelination into intrinsic and extrinsic neuronal mechanisms. Insufficient intrinsic growth capacity. So people like Kevin Park and Jay Lee are working primarily, and Vance Lemon and John Bixby and Hassan Ali working on these intrinsic growth capacity. And then the external inhibitory environment, that's a really big area in stroke, brain trauma, and spinal cord injury. And people like Jay Lee are working on the glial scar and trying to understand better the cellular molecular frame up of this glial scar, and does it really inhibit successful axonal regeneration? So again, to target axonal regeneration, we've used high content screening and evaluating many compounds. And this is the group, Vance, John, and Hassan, and they've isolated a series of small molecules that seem to enhance pretty robust regeneration of the cortical spinal tract. And that's the area that we've had a lot of trouble with in terms of regenerating. And they just got a $6 million grant from NIH. It's a Blueprint Neurotherapeutics Network grant that's going to allow them to take, hopefully, the lead compound all the way to an IND. So we're really excited about that. I should also mention that that Alan and Shelby Brooks and Aisha Khan just got NIH DoD funding for that for a study for peripheral nerve regeneration, which I'm going to get to in a second. So these are some of our clinical trials in the Miami project. We've gotten IND on human Schwann cell transplantation, Schwann cell derived bridging strategies with, again, Alan and others, brain-computer interface work, deep brain stimulation, mesenchymal stem cell treatment, neuro stem cell treatment, and now the next thing is we're moving forward is human Schwann cell derived exosomes for neurodegenerative diseases. These are the types of stem cells we can gather. We're using Schwann cells, as you know, for many reasons. You can take a peripheral nerve biopsy. You can generate millions of Schwann cells with a 97, 98, 99% purity. A lot of people are really interested in that. Aisha Khan. Aisha is one of the directors of the Interdisciplinary Stem Cell Institute. So she works with the Miami project very, very closely and processes all at our cells so we can put them into people. This is our first study with subacute spinal cord injury, showing that we can actually do this and we can do it safely, which was at the time a new experience for us. This is the study that I mentioned with Alan and Shelby and Aisha, looking at peripheral nerve regeneration, which is another major issue, especially when the gap is large. So a combination of this bridging collagen matrix with the seeding the Schwann cells seem to have a nice beneficial effect in a couple of subjects, and they just got funding, as I said, to continue this. And Dr. Brooks is working in the laboratory on different strategies to enhance that the good outcome. So I'm really excited about this. As I said, our next steps are taking exosomes from the human Schwann cells, or exosomes from the mesenchymal stem cells, or exosomes from neuroprogenitor cells, and seeing if we can inject these systemically and actually provide protection and repair. And this is the data. This is the NIH grant I just submitted. Plus, I submitted as the DoD. And it's showing a real severe model of traumatic brain injury. This is the penetrating TBI model that Ross Bullock and colleagues brought to the project years ago. And I said, well, if these have any chance of working clinically, we've got to do a really severe injury. And we had a nice Japanese neurosurgeon that worked with us for a year. And these are the data that we show. And basically what we see is that this is where the probe goes for the penetrating injury, but this is nice. We call it a penumbra, if you will. This is the area, it's inconsistencies, but you see, as you look at the data, you have a very nice protection in terms of the pathology. And we just replicated this in a second TBI model. I try to make sure I see a beneficial effect in 2 models before we really get excited about it. And a more conventional mouse model of controlled cortical impact injury that a lot of people use, we see a nice reduction in contusion volume. So there's something there in terms of neuroprotection, and we know in cell culture the exosomes also seem to enhance neurite growth in hippocampal neurons. So we've got 2 nice pieces of data, and we've done some preliminary work on the cargo. And as I mentioned before, the cargo is rich. So it could be doing a lot of cool things. So one day, maybe we can be injecting these cells and these exosomes and showing an effect. These are human. Because we just wanted to start with clinically relevant, approach. And we didn't immunosuppress. We saw no effect on lung. We saw no adverse effects. The animals lived for 24. It's just 1 injection, though. We just did 1 injection. So we did human. Yeah. Jugular. We infused them jugularly. Yeah. Yeah. So yeah. Yeah, we've been focusing on that because of the reasons that we know about. So anyway, yeah, we just saw human, and we had a short discussion with Damien about it. immunodepression, things like that. We said, no, let's just do this and see what one injection does. Ultimately, we'll have to do multiple injections. We might run into problems, but so far the literature says no. I mean, in the stroke world, they're doing a lot of exosome treatment now in ischemic stroke. This is some of the first data that we're seeing in brain and trauma. And Damian Pierce is, as you know, trying to do it in spinal cord injury right now. So looks cool. Okay, clinical translation. I've already talked a little bit about that. But this is what we're interested in. The Lens Center is opened up. I don't know how many people get over there and see the center. It's really beautiful, and we're trying to fill it up. It's filled up with patients, and we're trying to fill it up with subjects to go through some of our intensive rehabilitation exercise protocols targeting upper and lower extremity. This is our clinical research outreach programs, and we have about 20 active clinical studies: activity, nutrition, pain, spasticity, fertility, sleep, breathing, rehabilitation, neuromodulation, electromagnetic stimulation. So we've got a lot of things going on. There's a lot of quality of life issues that these people want us to look at. During COVID, we did a lot of that work at home. And in fact, one of our companies that we're working with, BrainQ, and is from Israel, they're actually allowing us to take some of the equipment home so the patient can do stimulation studies and rehabilitation studies at home. And we have this big educational program now that we're continuing after COVID. So we're doing a lot of neuromodulation, a lot of excitement in this field: motor training, robotics, deep brain stimulation, brain-computer interface work, transcutaneous stimulation, electromagnetic stimulation. So I thought it'd be a good idea to have a new institute at the University of Miami, and it's Institute of Neural Engineering. So about 4 years ago, we went through the process, and we have a nice engineering. And as you go on the first floor, we have about 4 biomedical engineers downstairs now. They're delivering fantastic biomaterials. We've got a nice study going on with Dr. Dumont and others. So that's a bright area. And we just got a new Bsms degree in neuroengineering. So a lot of the neuroscience undergraduates are signing up for that. My wife, Dr. Helen Bramlett, runs the undergraduate neuroscience program at the University of Miami. And so a lot of those individuals now want to sign up for this particular course. So I think, engineering, biotechnology, all of you are interested in that. So we're going to have the next generation of students, trainees coming into our laboratories. So it's going to enhance successful collaborations and infrastructure. And it's helping us build an infrastructure to accomplish this goal. This is BrainQ. We're doing spinal cord injury. We're doing our 4th patient now. And then they came to me and they wanted to do stroke. So we were working very closely with neurology, and we just recruited our first subject. And this is the BrainQ. They have different types of stimulation paradigms, that with the hypothesis being it firms up synapses, it enhances some circuit plasticity and things of this nature. So that's moving forward. The Uplift study you've heard about, this is transcutaneous stimulation. They've been the prototypes we've used for a number of years. And now this is a 14-center, multi-center study that just got completed. Jim Guest was the site PI for this. And the results are kind of amazing. I mean, we only did 2 subjects, but it blew me away. Somebody that I've known for 30 years that could not really move their hand can now move their hand. And the other subject too. So this neuromodulation, targeting these residual circuits. So I think we've got this this formula where we can do a lot of things, and then at the end we can do the neuromodulation and really enhance some of the benefits that we're seeing with the other therapies. This is Patrick Ganser. He's our new recruit. It's a dual recruit. It's my first dual recruit with biomedical engineering and neurological surgery. And he does a lot of vagal nerve stimulation. And we've got a company in Texas, and we're just starting to firm up the clinical trial for that, looking at vagal nerve stimulation, looking at upper extremity function, for example. And as you know, the vagus nerve really controls a lot of things, the autonomic nervous system. So we're also looking at some autonomic dysfunction in this patient population. So this is going to be an exciting trial next year. So the project, with the help of neurological surgery, is a holistic approach targeting different phases of injury and repair, We've already talked about the neuroprotection, pharmacotherapy, the acute neurosurgical procedures, hypothermia, trying to promote endogenous reparative processes. Interesting about the viruses you talked about now having an effect on stem cell proliferation, things of this nature. You have to think about that a little bit more. Developing novel bridging strategies. And again, the medical engineers downstairs think about this all the time. Innovative cellular transplants, targeting growth inhibitory factors, improving axonal function and neurorehabilitation, retraining, enabling the injured brain. And then on top of that, you put the engineering part of it in terms of nanotechnology, tissue engineering, neuroimaging, diagnostic approaches, human motor and sensory control, and of course, brain-computer interface work that we're very interested in pursuing as well. We thank the Bone County Fund and other funding agencies. We write a lot of grants. This month I've sent 4 grants in, a couple to the NIH and a couple to the Department of Defense, and everybody does that. So funding is good. It could be, we're really trying to focus on NIH funding. We got a couple of new NIH grants this month, and that's going to help bring the Department of Neurological Surgery, hopefully to the top 10 in terms of NIH funding. We got to number 4 at one time, I think. But it's not that we're losing funding, it's that all these other places are increasing their funding. So it's always a moving target, if you will. But we thank you all for your contributions to what we're trying to do. We have a great team. We work with many clinical departments and centers at the university. And again, I think this is a special place where discoveries are transformed into breakthroughs and therapies, hopefully. So with that in mind, I hope all of you are interested in what we're doing in this building in terms of some of the preclinical work. And, you know, we're building alliances with many clinical departments. And so if you have any ideas about collaborations or clinical translation, please come talk to us about it. We're excited about what you guys are doing and how we can do things together. So thank you very much.
Status of hypothermia for TBI for, we're doing that trial. We talked about it here.
Yeah. Yes. So the HOPES trial was getting at, heterogeneity of the TBI population. That, when a neurosurgeon goes in and decompresses, and now you have all this warm blood going into the ischemic zone or the contusion zone. If you could cool that particular area, would you actually target reperfusion injury and improve? So that was with, I know when I came home, I heard this talk. It was actually, I think, myocardial infarction that I got the idea about the reperfusion injury. So I came and talked to Ross Bullock and said, don't you have a model of, subdural or something? He goes, yeah. So we brought it out, we tested it, and it worked. And so we started the clinical trial, and it was difficult in Japan, in the United States, to collect the patient population. We never got a critical mass of people recruiting these subjects. And so we couldn't really do the study. So it kind of just kind of went away. But with the stroke now with thrombolysis, these catheters now are delivering cold saline at the same time they're actually capturing the thrombus. And that looks really cool. With Bobby Starks, we've published now with 3 papers now, where if you take that thrombus out of the patient and measure inflammasome levels, they're sky high. So that thrombus is not just occluding a vessel, it's releasing pro-inflammatory cytokines into the circulation. So that's what I've always emphasized to people that listen to me, that stroke is not clamping a vessel. Stroke is, obviously ischemia, but it's a lot of different types of things going on in the pathogenesis.
Yeah, I ran into Ryan Kitagawa, who was a fellow back in the days when this project was being created, and it was a collaborative effort between Miami and Memorial Hermann. The chair at the time was one of the people who got the initial funds to start it, and basically it's over. I think maybe 10 patients got recruited. They published the paper apparently in some obscure journal. I've been meaning to.
I'd have to find that. I haven't seen it either.
Yeah, and I've been meaning to pull it up. I was wondering, when Christy was here, we talked a little bit about hypothermia and myocardial infarction and neuroprotective effects. And out of hospital cardiac arrest and in hospital cardiac arrest. And she was saying it's still within the protocol. I was just wondering if that was discussed in your recent European meeting. I'm sure that was sort of headlines at that point.
Yeah, we've got the TMM 1 and 2 clinical trials that really made us start thinking about the critical temperature in cardiac arrest patients. And everyone kind of agrees that, getting it above, 37.6 is bad for the patient. So everybody's doing their best to make sure the temperature doesn't go above that. But in terms of the level of cooling, it's up in the air right now. So I gave a talk basically starting with the discovery research, showing how important small differences in temperature were in a model of transient global ischemia, which kind of mimics the pathophysiology and the cognitive effects of cardiac arrest, and showed that there was a big difference between 36 and 33. And so, and then the next part of the study was basically showing the new data coming out since the TTM1 and TTM2 studies have finished. Many people just threw out, targeted temperature management in their clinical treatment protocols, and mortality went way up. And it's clear now. It's paper after paper showing that if you don't do targeted temperature management, you don't get good results in big patient population studies. So now they're arguing exactly how we just decide a patient should be maintained at 36 degrees, or should a patient be maintained at 35 degrees, or should a patient be maintained at 33 degrees. And basically, it's all about some type of biomarkers and strategies to triage the patient. Maybe it's hard to do it immediately, but maybe at 24 hours, you can do some biomarkers or EKG or something that will tell you exactly how the patient's doing. So that's the big area right now, is actually what the temperature. They're not throwing out targeted temperature management at all. They're just trying to figure out what's the best temperature. And people, a lot of groups say, well, I do 36. I mean, I'm always going to do 36 because it's just too much trouble to do the 33, right? If you don't have to do it. The other groups say, no, we're all doing 33, 34. And so it's different populations, and that discussion is fine. It'll continue. But, if you ever see me come in a cardiac arrest, put me down to 33, okay? All right. Thank you very much. Take care. Have a good day.